Okay, so let's talk about microarrays a little bit. This image here is actually a microarray, or supposed to. This is a cartoon picture of a microarray, and it's actually a really cool technology. It was really a big cutting edge deal when I was starting here as an assistant professor, and a little bit before that. And so I got into microarrays after being an entomologist. So I wasn't really much of a molecular biologist, but I learned quite a bit just so I could do this, add this to my toolbox. So one of the things I recommend as a scientist is to always keep adding to your toolbox so that you're relevant. I hear sometimes people say, "Well, I'm an ecologist. I don't need to learn molecular biologist," or molecular biologists seem to be ignorant about the organism. You really, if you really want to be that scientist that stands out, you need to take time to learn and appreciate both fields, at least from the perspective of molecular biology being a tool, so that you can use it in your research with organisms. Or if you're really a molecular biologist and you get off on that, you need to think about the organism and understand it. Because I know a lot of people that have the toolbox but don't have any questions to answer, or at least be able to communicate when you develop your collaborations. Now, again, when we talk about gene expression, we're talking about, and this is, so microarrays is a method to measure, one of the methods to measure gene expression. It can be used for measuring other things that are molecular, like DNA. But gene expression means messenger RNA. So remember, DNA obviously is in the nucleus of cells. When it transcribes, it makes a segment of messenger RNA. I like to tell students, especially in my freshman classes, that DNA is the blueprints that all of you should know that already. They're like a big book of information on how to make proteins that it's inside your cell, inside your nucleus, of course. So messenger RNA is like taking a photocopy of a page of that, sending it out into the cell's extracellular matrix, where it hooks up with ribosomes, and then that photocopy, that messenger RNA, is then, of course, used to make proteins. We call it the central dogma in biology. And so whenever I talk about measuring genes, I don't really mean measuring genes, I mean measuring gene expression, just to make sure you understand that. And again, measuring gene expression is measuring the amount of messenger RNA transcripts for any given gene we're interested in. So let's say I have my caterpillars feeding on a plant like tomatoes, and it starts wounding the tomato plant. It's going to raise messenger RNA levels for protease inhibitors. Remember, protease inhibitors is that anti-nutritive defense. And so this anti-nutritive defense is going to keep building up and building up as an actual compound, but before it gets to be an actual compound, you have to have lots of these messenger RNA transcripts, photocopies being made and sent out into the cell to make the actual protease inhibitor. So we want to measure how many photocopies of protease inhibitors are being made, and that's what we're talking about, what I mean by gene expression. In the old days, what they did is a thing called northern blots. So northern blots was to take, it could be done in different ways, but basically we had purified RNA from a cell, total RNA. Now remember, your body or your cells have three different types of RNA. We have messenger RNA, which is what we're interested in measuring. We also have transfer RNA, which, if hopefully you remember this from a general biology class, it's important for bringing amino acids over to make the protein. And then we have ribosomes, which are also called ribosomal RNA. So ribosomes are made up of this ribosomal RNA. I may obviously have some proteins and things like that too. But when you purify the RNA, and this is something I would do in this class, so if somebody is really interested in that, we might talk later. But we would take our plant or take our caterpillar and purify all the RNA from it after giving it these different treatments, whether it be feeding it nicotine or letting the plants feed or caterpillars feed on the plant. taking that plant and taking on the RNA from it. So that was something we did quite often in this class. So after we purify this RNA, we would run it on a gel in the old days. And then we would label for that RNA specifically based on taking some of the anti-transcripts or something like that that's with a colored dye on it that would glow. In fact, I haven't really done northern blots myself. How did the name northern blots come up? This might be kind of interesting to you, though. When we started getting into, this is again more of a molecular, describing how a little bit transcendental, but when we talk about molecular biology, some of the first things that were ever purified was DNA. And then they would run it on a gel. So again, I'm hoping you realize a gel is kind of like this little matrix of gelatin that you run it through using electricity to draw through the gel based on size. Well, the person that first invented that, his last name was Southern. So when you did Southern blots, that was for DNA. So what happened next is when somebody figured out how to do it for RNA, they just decided to call it Northern. So actually Northern is kind of a play on the guy's previous last name, Southern. They just started calling it Northern blots for RNA. And then you may have heard of Western blots. So so now you're starting to see the plays on north, south, east, and west. So Western blots is measuring protein directly. So anyway, when we did this, it was a very laborious, intensive technique, and you do radioactive detections and all sorts of stuff. The only person I really know of that really did it in our school right now is Dr. Hummusser, or Hum. She's the one that was very involved with this back when she was working on her degrees. So what you see here is a gel, and it would be ran across, and then you would take a probe that would measure that RNA. So these are just the size of this would represent a gene of interest. So let's say that looking at gel A, this is a gel. So again, it'd be like gelatin. And then you'd have a well and you'd fill it up with your RNA or DNA. It'd run across, and then you would take a probe that would be radioactive or whatever that would stick for that particular type of gene, and it would glow. And so the bigger the bands were, the more of that RNA was present. So you can see in this particular lane 4 that it wasn't present in this hypothetical treatment. While this other one's, it was really big and bold, so that means it was being stimulated. Again, this is all being kind of a hypothetical based on these different experiments. Well, I guess it wouldn't be hypothetical, but then those experiments. And here's another gel running the same particular RNA. So there's different ones that they were probing for. So that means there's other ones in here, but you just don't see them because it wasn't probed for. Now, everything was measured against a control, a housekeeping gene. Now, this is kind of important to keep in mind. A housekeeping gene is a gene that's always turned on, in quotes, always turned on, irrespective of the treatment. And that allows us to know that we're using the same amount of RNA. So let's imagine that, again, going back to the idea of we have these two different treatments. Let's just imagine caterpillars with a normal diet, caterpillars with a toxin like nicotine. And then we purified the RNA from the caterpillars. We would need to make sure that we were using the same amount of RNA from each of these caterpillars, and we weren't artificially messing up our results by taking a lot more RNA from this treatment than this treatment. And so the actin down here is an example of a housekeeping gene. And so they would run that as well, and they would see, oh, look, we used the same amount of RNA across all 4 of these treatments. These 3 treatments, these genes showed up really big and bold, but in the lane 4, that treatment, it didn't show up. Or if you go over here and look at these replicates over here, if they're comparing these 2 different gels, you can see it's not showing up. So this was their way, again, of trying to show that this RNA was important for this particular treatment. To make a long story short, it was a very laborious, time-intensive situation. You could spend 24 hours doing this. Imagine if we could come up with a way where we did thousands of these, but just a one-time pass. And that's what the beauty of microarrays are. Instead of measuring one or handful of genes over 24 hours, you can literally measure thousands or tens of thousands, 20,000, 30,000 genes at one given time. So that's what these microarrays allowed us to do. Again, a lot of this came up, I believe, in the 90s, late 90s, looking at yeast. And I'll show and explain that a little bit in a moment. So again, microarrays were developed by the availability of large databases of cDNA. So cDNA is basically a copy through reverse transcription of a gene. So remember, messenger RNA is made up of RNA, and then we can reverse transcribe it and make cDNA. And so if you're familiar with, I'm hoping, again, you should be familiar with transcription and translation, and of course you should be somewhat familiar with reverse transcription, where if you had, let's say, a sequence of genes like AUG, that would represent the start codon for messenger RNA, AUG. I'm hoping these nucleotides make sense to you. You would just find the reverse of that to make reverse transcribe, and that's what we call cDNA. Does that ring a bell a little bit to everybody? So what it would be, the reverse nucleotide for A, it would be T. What about U? It would be A, right? And then C and G and all that kind of stuff. Okay. So anyway, I hope you're familiar with that. But the point is we have these huge databases of cDNA for lots of organisms. And then the technology has gone even further than what I'm describing here. This is kind of like part of it's like the Human Genome Project. We started sequencing large groups of genes for organisms that we could then use to analyze. And then lots of people studied various genes, and we basically developed these huge databases. And so anyway, it becomes this big library that then we can basically perform experiments like what I described, where we treat caterpillars with a toxin, without a toxin, and literally look at thousands of genes and whether or not those are turned on, being transcribed, and so forth. Okay, so as I mentioned before, doing the northern blots was pretty laborious, intensive, time intensive. We now do quantitative real-time PCR often if we have that kind of machine, or some variant of real-time PCR, and we also do microarrays. And so again, the ability to do microarrays was the fact that we were able to get into sequencing on a very large scale and have these databases, and we have an idea of what each of the different genes, and we can make probes, and we can look at all sorts of different physiological treatments and tissue topologies and histologies to do this. So essentially it's allowing us to look at the gene expression of thousands of genes. So it's basically a massive parallel kind of experiment, literally doing thousands of experiments. I kind of even mentioned, if you remember from previously, when we deal with stats, it starts to become kind of hairy when you're dealing with thousands of experiments at one time, trying to figure out whether something is statistically significantly different in a microarray experiment. But that's more for maybe another time or detail or discussion later maybe. But again, back in the late 90s, a lot of this was being developed by looking at yeast initially. And so they would make these huge libraries of cDNA looking at the genes of yeast, and then they would print it onto these slides using kind of a quill pen-like setup or robotic setup to put these spots on it. Each one of these spots would represent some of this library of probes for a particular gene of interest for a particular spot. And I'll show you a picture of that. But if you can kind of look at this glass slide here and then zoom in over here, you can kind of see these boxes. Well, each one of these boxes is made up of little dots of genes of interest. And it got to the point where the technology got so crazy cool that it actually could almost be like, instead of a pin putting the spot of gene on, it could actually be sprayed on like a inkjet printer. But let me show you how it was done back when I was finishing up my PhD and starting a postdoc. It was done by this kind of big robot. Well, I'm calling it a robot, but it's basically a computer-controlled arm that goes back and forth, and it was protected from the environment in the box. This was actually one in the University of Arizona. So there's little pins, and it comes in and washes off in this beaker of solution that helps clean the pins, and then it goes over to here, and you can't really see it, but it's like 96 well plates containing probes of genes of interest, and it'll suck up some of the probe, come over here and print it onto glass slides. You can't make it out very well, but these are tons of glass slides in columns, and here's the rows for them. And the robot will just come around and print it onto the slide. It'll just come along, suck up some, and just kind of tap it along. These are actually safety buttons. So if you get stuck in the machine, you don't want to get printed on, or there's something dangerous going on, you can actually hit on them to stop the machine from working. Here's another animation kind of, well, picture an animation of it in action. I don't know if I can print on it and see anything happen. So, but these are glass slides. This would be the pins. This is the robot arm going back and forth. It would suck up the genes of interest out of these wells through, and again, these have been through PCR, but amplified. Hope you remember a little bit about PCR. But basically, a gene of interest would be sucked up a half sequence for the, it would be a sequence we were interested in, and then these would be glass slides that it would print on. And here's another picture to help you see the pins. So here's again the robot arm that's going back and forth. And then here's the pins. And if you zoom in on the pins, you'll see it looks kind of like a quill pen. If you remember the old-fashioned quill pens that made from a feather, and then you can suck up ink. Well, that's kind of what's happening here. It's sucking up the probe of interest, the gene of interest, and then it comes along and just taps on the glass. Basically these pins just come along and tap on the glass, laying down the genes of interest, and it'll go wash itself in this well of water here. And so it'll actually leave a little droplet, and that droplet will stick to the glass of a gene of interest. And you can literally have rays. So that's why a microarray, it's an array, a long line of genes of interest. So if you look at this slide again, it looks like there's a bunch of boxes lightly covered. Well, on it is lots of small little spots representing a gene. So it is a microarray, and it'll stick to it. And so then you can do your treatment. So like I said before, you can hypothetically have a virus treatment versus a non-virus treatment in plants, or plants fed on with caterpillars or without caterpillars. You can look at cancer cells versus healthy tissue. You'll purify the RNA from it. That includes all the RNA, and then you ultimately usually purify or get the messenger RNA out of it and label that specifically. And there's all sorts of ways to do this. It's just one of the many different ways. But you basically label the messenger RNA with these different colored dyes that represent these different treatments. And so maybe this is the virus treatment, this is the non-virus treatment, or this is the cancer treatment or the non-cancer treatment. It doesn't have to mean that the treatment is red and the artificial treatment is green, but that's just one of the ways it could happen. You can flip them around, and we do things called dye reversal because we try to avoid dye biases and stuff. But this is all done through reverse transcription, at least one of the methods. There's other methods where you purify the RNA from your tissue samples, and then through reverse transcription, you make complementary DNA. Remember the cDNA? So you take that RNA, that single strand of RNA, and you have your different letters for your different codons, and then the reverse transcriptase enzyme, and you'll have a bunch of nucleotides in your solution, and it'll go along and make a sequence. So if you had a bunch of T's, what would be the corresponding letters to that? A's, right? Because the A nucleotides, so you have a lot of thymine nucleotides, you're going to have a lot of adenine nucleotides. Actually, eukaryotes, remember, cells with a true membrane, organelles, and nucleus, those are eukaryotic cells versus bacteria. Well, their messenger RNA can also has a long poly A tail. We call it poly A RNA also. So when we do reverse transcription, you'll see a strand of T nucleotides, and I'll show you that in a moment. But anyway, bottom line, if you're confused about everything I said, we're labeling one of the treatments red, one of the treatments green from the RNA or the cDNA, depending on how you make it from these two different treatments. And then we can put it on a slide for one of those genes and then see whether it shows up red or green. And again, if it's present in high amounts, you'll see that. And I'll show you a video that helps you to understand that in a minute if I'm confusing. So basically, you've done these two different treatments. You've purified the RNA, labeled it, so forth and so on. Then you put it onto a slide. That's what's happening here. It's both of those treatments are put together into one microcentrifuge tube. You suck up the solution, and then you put it onto a slide. And then usually you just put a cover slip on it, because remember, it's a microscope slide, special microscope slide that's been treated with these microarrays on it. And then you put the slide on it, it actually covers that film of genes and water. It's water. It looks like water, right? And it is water in there too, but it's going to have that labeled messenger RNA. And then you just let it float around for a while, and then it's going to stick to its complementary nucleotides. And if it doesn't stick, it's going to just be washed away. We call that hybridization. And so this falcon tube is our hybridization chamber, in quotes. So it's just going to be kind of a paper towel that's a little bit moist to provide a little humidity. And usually you keep it around 55 degrees. Well, no, I don't think it's that hot. But you keep it at a reasonable temperature it's above room temperature until the probes and the targets stick together. And so this would represent the glass slide, and these are the probes that are already on there. So here's our nucleotides representing a particular gene of interest. So it's going to be a critical part of that gene. And then our target cDNA or RNA, because actually you can do RNA too, will be labeled either red or green, and it will stick to those available probes. And the relative amount of it will represent how much gene is present. Anything that doesn't stick, the next day we take this slide out and wash it, and then we run it in this microarray scanner. It's a laser scanner that scans red and scans green and sees whether or not it fluoresces based on our labeled cDNA, complementary DNA. This is one of the old school microarray scanners. Like I showed you, one in my lab is actually blue, but you can put the slide right in here and the lasers will go to work and give you an image for your computer. It tells you the intensities and so forth. So it'll scan green and it'll scan red. We'll get these spots, and then the computer actually gives it a false color overlay. So it turns all these spots to look green and all these spots to look red, overlays these two images, and you get the relative green to reds. Now remember, in our example, the greens might represent the control treatment and the red represents the infected treatment. So anytime you see a spot, this red would represent, that gene being turned on because of the virus treatment, and the green would represent it being turned on in the control treatment or suppressed by the virus treatment, just depending on how you look at it. Or if you're talking about cancer versus non-cancer, the reds would be genes would be turned on by the cancer treatment, greens would be turned on by the control treatment, and if you see yellows, that means they're being turned on by both. And then there'll be lots of spots that just aren't turned on by either treatment. They're not important for that. Here's another picture kind of showing you this relative amounts. So again, each one of these spots would represent a gene of interest. And so this would be a gene that would be turned on, by our example, by the virus treatment. This gene would be turned on by the control treatment. While these genes wouldn't be turned on at all, even though there's spots there, they're just not being labeled. And then again, if it's yellowish, that usually means that it was turned on by both treatments equally. So in a sense, it wasn't, the treatment might not have been considered important. So what you do now is you can massively screen 30,000 genes and say, all these are important for the virus treatment. All these are important for the cancer treatment. See how powerful that is to be able to take the human genome and then figure out which genes are being turned on because of this cancer treatment. Or look at the tobacco plant. These genes are being turned on by the caterpillar spit. So that's what makes this powerful. Now, we usually consider this a kind of, while it provides good evidence and you do statistics, usually you will still want to have some type of confirmation, either through quantitative real-time PCR or some other method. Now, this was it was really a great technology, still a very valuable technology if you have the setup. And now people are moving to next generation sequencing, which allows you to sequence and do your experiments in parallel and is considered very powerful. Usually next generation sequencing is a term that you'll hear a lot more nowadays, is the way that a lot of people do things. It's basically sequencing on the fly, if you want to put it in simpler terms. So again, this is kind of how a microarray setup is done. Somebody has sequenced, like the Human Genome Project has sequenced a bunch of genes for a particular organism of interest through different treatments. And that's what the DNA clones represent. So those will be then put onto a chip by one of those robots. So it's printing the arrays, the microarrays. Then you do your experimental treatments. You'll have your control treatment and your tested treatment. And it doesn't have to be red or green necessarily for your test or your control. That's up to your decision. And often I recommend doing a dye reversal where sometimes the test treatments are red, sometimes the test treatments are green, and vice versa for the reference or the control treatment. So you'll collect the RNA from your samples, do reverse transcription if you're doing cDNA, microarray chips. Again, there's a few different ones. While you're making it, you will label it red or green. So there's actually nucleotides with the dye attached to it. So you remember the nucleotides of DNA or RNA, it's the A's, the T's, the C's, and the G's. Well, I believe the C nucleotide has the dye attached to it. And so it'll either be red or at least it'll fluoresce red or green. The interesting thing is, even though it looks red or green in the test tubes, they actually fluoresce the opposite color, but don't worry about that so much. We put it on this chip, we hybridize it. Remember that little chamber, the Falcon tube? We hybridize it there, and then we'll scan it red, and then we'll scan it green, and then we'll have this computer image where we put everything on top. And if it's more red, that means the test treatment was turned on. If it's more green, the control treatment was turned on. How did I get into it? Well, I visited this lab in Switzerland. I'd gotten into, remember how I cauterized the spinner out of caterpillars so they couldn't secrete saliva? Well, I told this famous researcher, Ted Farmer, about it, that, hey, I got this surgery, and you got this technique to measure plant gene expression. Can I come out and visit? So me and Sue went out to Switzerland and visited his university. The university is kind of spread out. They got all these buildings everywhere. There's a lot more buildings than it looks, but this is actually their science building. That they have other parts of the campus too. They even have they had even plants growing on top of the roof as part of their kind of ecological, good for nature kind of approach. And this is University of Lausanne, Switzerland. And even they used animals to help keep the grass mowed, so to speak, without, again, the idea of being good for the environment. And the nice thing about this kind of school, and maybe you can find it in other places too, but the lab becomes like a family. So the researchers get together often. At the end of the week, they would have wine and cheese. They'd often take a coffee break together in Switzerland. But he was one of the first to do a study on microarrays, looking at caterpillars feeding on Arabidopsis plants. So again, I sold the idea, hey, I'd like to come out and do my surgery on your caterpillars, and let's look and see what happens to gene expression. So again, I tried to do the cauterization out there, but it didn't work very well. So that's where I developed that surgery to remove the salivary glands from the caterpillars. So I removed the glands, let them float in the water, removed them, and they healed, and then they could go on and do their thing. So here I am doing the surgery and then putting them on Arabidopsis plants. This is Pyrrhus rapier is the name of the caterpillar. So we did the experiment. We had caterpillars that with salivary glands and without salivary glands feed on these plants. We collected the total RNA, that means ribosomal RNA and transfer RNA, but that's not what we're really interested in. We're interested in messenger RNA. The carbon, we called it the copy from the book or the instruction. And so we labeled the RNA through reverse transcription. We made cDNA using reverse transcriptase. Remember, there's a virus that has that enzyme. So that's where we learned how to do this, from those viruses. And then we label it either with the Cy3, which is the green dye, or Cy5, the red dye. We put it on the microchip, and then we looked at the treatments, and we found that the plants did turn on different genes, whether it was fed on by caterpillars with intact salivary glands or ablated salivary glands. A handful of genes were altered because of it. So this is how I kind of got into microarrays a little bit before coming to Western. I also did a postdoc at the University of Arizona looking at corn plants. That's because that's what they were interested in. So here I have caterpillars feeding on corn plants at the University of Arizona for my postdoc. This is typically, need to do a postdoc if you want to get a position at a university after you complete your PhD. And so that's where I got into microarrays more. So I found that there's a lot of genes that are altered because of the caterpillar spin, particularly drought-resistant genes. So that's how I came to Western and kind of got into all of this. Let me show you a quick animation on this topic that I think will help to explain it a little bit further. So this is a lot like the animation I wanted to show you. And so again, we're going to be talking about DNA microarrays. There's also RNA microarrays. There's also, it's a lot more advanced than when I first started. And it got to the point where we could go from a few hundred genes or a couple thousand genes in a plant to looking at literally 50,000 genes and all sorts of things. It got pretty crazy. But again, this is representing that array. And so you got these different spots on them. The red spots will be turned on for one treatment. Green spots would mean it represents another. Yellow means they're both equally stimulated. If you don't see a spot, that means it wasn't turned on at all. Again, a lot of this was invented in yeast. So again, yeast. So here's again an example, basic research in yeast. A lot of people that are non-scientists might think, why are they wasting money on this science, this basic research science? But all these amazing things come from things that you might not have realized is important. So like even how we do some of this technology and turning off genes and stuff like that was discovered by looking at viruses on petunia plants. And now it's could have a major impact on medicine and molecular medicine and stuff like that. So you never know where some of the most crazy, cool scientific ideas will come from for helping humanity, and not just, of course, the fact that it's already cool to begin with. So,
reveal the expression of thousands of genes on a solid surface, such as a microscope slide. In this example, we'll use yeast as a model system to illustrate one.
Now remember, what's cool about yeast, or what's the deal about yeast? Well, yeast is aerobic and anaerobic. It can grow in both conditions. If it grows in anaerobic conditions, we get what? Well, we get alcohol and beer and stuff like that. If it gets anaerobic conditions, we have bread. But it turns on a different set of genes when it's in the aerobic condition versus the anaerobic condition, or the unoxygenated condition.
Use of microarrays. One common use of microarrays is to determine which genes are activated and which are repressed when two populations of cells are compared.
So again, here we got our yeast cells growing in an oxygenated environment. Imagine putting them in a test tube. And then we put them in another test tube, but we close the test tube over here so it doesn't have oxygen. It's going to return on a different set of genes because the metabolism is quite a bit different in an aerobic condition versus an anaerobic or non-aerobic condition.
Every gene is measured simultaneously. As an example, we'll compare what happens to yeast genes when cells are grown in aerobic versus anaerobic conditions. The cells grow and adjust which genes need to be activated or repressed in order to survive. Now it is time to isolate the mRNA from both populations of cells. The cells are spun in a centrifuge. Now that the cells have gathered in pellets,
So we have the pellets, and they're going to oversimplify it, but this is when you begin to purify the RNA from the treatments. And they said they jumped right to messenger RNA. Normally you purify the total RNA first, but there are kits now that you might be able to purify the messenger RNA directly.
we remove the liquid, but not the cells. Next, it is time to extract
Remember, the liquid is just the broth that they were growing in.
the mRNA from the cells. When we add the extraction buffer, the mRNA is released into the solution. Next, we remove the RNA and place it in a fresh tube.
And you see how they're always changing the tips out.
Now, let's make the cDNA from the mRNA.
Okay, so remember, yeast is a eukaryotic cell, single-celled organism, but it is eukaryotic. It has a true nucleus, true organelles. So their their messenger RNA is called poly-A RNA. So this is a poly-A tail. So these are A nucleotides. And then there's other nucleotides that are going to be present here as well. Since it's RNA, we're going to have AU and G and C. We're not going to have T's. Then we're going to do reverse transcription. Did I remember that right? I'm messing it up. It's been a while since I thought about this. But anyway, we're going to do reverse transcription, which means we're going to make cDNA coming off of it.
Here we see 3 of the many mRNA molecules from each tube of cells. Each
So again, this is the anaerobic and this is the aerobic.
mRNA is converted into red or green colored.
So we're going from the 3' end to the 5' end, and then you'll see that what would have been in this solution that isn't talked about is there's free nucleotides in here, plus nucleotides that are labeled red or green, and those are usually the C nucleotides.
cDNA. When the colored cDNA is made, the mRNA degrades.
So we used reverse transcription to do this, through the reverse transcriptase enzyme, and then we degrade the RNA. So we'll break it apart, and then destroy it with RNases, enzymes that break the RNA down.
Then we combine the red and green cDNA, mixing both colors into a single tube.
So again, we labeled them separately, and then then you can combine them, because at that point, the anaerobic and the aerobic conditions have been properly labeled, so it doesn't matter if you mix them after you've made that cDNA.
At last, it's time to look at the DNA microarray. In our experiment, a microarray or DNA chip contains about 6,000 spots. Each spot is a different yeast coding sequence from a different gene. Let's choose 3 spots at random to follow in detail. Each spot is made of DNA that can base pair with its complementary cDNA. Here are partial sequences from each of the 3 spots we are observing.
So again, remember the probes are going to be on there. They're just picking a few letters from it. G, A, T, and C, and so forth. It would be quite a bit longer than that. And then here's another gene. And again, these are going to be the unique sequences of that gene. So those, again, are the probes that are on out of the 6,000.
Observing. Now, let's incubate the mixed cDNA with the DNA chip.
Incubation is the same thing when I said hybridization earlier, when we put it in that falcon tube and close it with the humidity. That's usually an overnight process versus floating around, and then it's going to hook up with any nucleotides that match it.
For the sake of our example, we'll zoom in and show that some of the labeled cDNA have bound to the DNA in the spots and formed base pairings. Here we see green and red cDNA bound to this spot. Only red cDNA is bound to this spot. And only green cDNA bound to this other spot. In a real experiment, you would not see any of this detail. You would only see the original microarray. Now we must wash off the unbound cDNA to see what is bound to the microarray. Let's detect the bound cDNA so it can be visualized. We begin by placing the microscope slide containing the microarray inside a scanner. We'll examine the next phase of the process, keeping our focus on the 3 spots we've been following. First, a green laser scans the microarray.
So you notice how out of those 3 spots, 3 started to glow and 1 isn't. And again, I believe that green is the aerobic condition.
The resulting image is stored on a computer for later analysis. Now it's time for the red laser. This image is also stored on a computer for later analysis. Now, we move to the analysis phase. After we eject and safely store the microscope slide, we retrieve the red and green images from the computer and create a merged visualization.
So remember, I showed you a big image where we have these different colors. In this case, this treatment was really turned on in the anaerobic condition, the red, and the green. This was obviously important for the aerobic condition. You could argue it's being suppressed or repressed. And then this particular gene, it's turned on by both of these treatments. It doesn't, in this case, that gene is being turned on irrespective of the treatments. And when I say turned on, I mean gene expressed. The messenger RNA was being made.
In the merged image, we see an aerobic gene labeled in green, an anaerobic gene labeled in red, and a gene labeled in yellow that was expressed in both aerobic and anaerobic conditions.
The one thing I will say, you don't really ever say only. It's all relative. So that means there's probably some red in here, and there's probably some green in here. And you get to that finer detail when you scan it and you're looking at intensities. So it's a little bit misleading. There could still be some background levels. So you might say it's 5-fold higher for this treatment versus that treatment. But she's just talking about it in a qualitative state.
This is one example of how DNA microarrays are used. In an actual experiment, quantitative analysis would be conducted on all 6,000 genes.
Oh, just like I said. Okay. So anyway, this led to lots of theses that were done in my lab, lots of theses that need to be published still. It was a credible challenge for me to kind of go from one extreme where I looked at nicotine to jumping and using this new technology. And particularly most of it, my success was actually at Western, even though I learned a little bit in Switzerland, and I learned a little bit in Arizona. I ended up getting with Sue getting a big grant called the Plant Genome Grant that allowed us to get a lot of this stuff from National Science Foundation. That was also another struggle, which took about 7 years of work before I actually was successful enough to do so. All right. So does anybody have any questions? I don't know if anybody's really particularly confused. In the end of the day, just realize it's a massive way to screen through thousands of genes. And it gives us it's a really great tool. And again, it would take a while to really learn this. I just hope you get at least some understanding now. All right, so I don't hear any questions. Is that correct? I'm going to stop recording now.
No.
Okay, let me stop recording.